slides. Two just things I want to mention before I get going. One is I'm going to do a total of 5 lectures. Just the timing of the Zoom lectures and the fact that I've added some material, I'm going to do one more lecture than I have in the past. This is number 3. There will be 2 more. They will be on successive Wednesdays at 2, this time, next week, same place. Secondly, let me just say that of the 5 lectures, this is going to be the most biochemical of the lectures. But the good thing is, when you get to the end of this lecture, and we're going to address the question of how do you actually get energy from your food, and what does that mean? You'll know more about this than 99% of the people out there, all right? So it's going to be a little bit chemical, but it'll probably be pretty much new material for many of you. So last time, we talked about sugars, and the most abundant sugars we eat are sucrose, and that's converted to glucose and fructose, so we gotta get a lot of glucose there. We eat starches. Starches are just chains of glucose, and so perhaps the single most abundant molecule in our food is this molecule glucose, and it's a central molecule in the metabolism of our food. And we breathe in oxygen, we somehow combine it with glucose, or do we really? And we drink water. We have to have water to make this process go. And the result is that we end up building the molecules in our body, and we use most of it for energy. And what does that mean anyway? All right, so we breathe in oxygen, we breathe out CO2, and we breathe out water vapor. So what happens to the oxygen? How do we actually use it? Where does the CO2 actually come from? Is this oxygen from the oxygen that we breathe in? Well, it turns out no. Where does the water vapor come from? Is this the same water vapor that we breathed in? No. And in what form is the energy? Okay, so let me go here. So to understand this, you have to know how enzymes work, because enzymes are what drive all of these chemical reactions in our body, in all living things. The reactions, the conversion of one molecule to another, the building up of complicated molecules, it's all done by enzymes. So let's talk about what they are and how they work. So in this little cartoon here, we're going to have this big purple one. That's an enzyme. I'm going to show you more details about its structure. And let's say this is a molecule of sucrose, that's simple sugar. And last time we saw that sucrose gets split into 2 sugars to make glucose and fructose. So the shape of glucose, or sucrose rather, can be exactly mirrored by the shape of the surface of an enzyme molecule, such that the sucrose molecules bouncing around here will bounce into this pocket, and once they meet there, the atoms on the surface of the enzyme interact with the atoms on the surface of the sucrose, and you get a chemical reaction. In this case, it splits it into 2 simpler sugars. And this is basically what enzymes do. They're not themselves changed in the chemical reaction at all, but they make the reaction go faster. So all enzymes, almost all enzymes, are made up of proteins. There are few tiny exceptions of enzymes that are made up of RNA molecules, but we won't go into that. And like all proteins, they're formed by long chains of amino acids. This is an amino acid down here. Typically, they'll have 500 to 1,000 amino acids. And since a typical amino acid has 10 to 20 atoms in it, like this one here, the 1, 2, 3, 4, 5, 6, 7, 8, 9, plus a side chain, about 10 amino acids. A typical protein is a very complex molecule with as many as 10,000 or more atoms. Okay, so one way to cartoon what a simple protein would look like. This is a very small one. This is a sequence of a real protein. It's a protein that chews up RNA. Is like it's a chain of amino acids. In this particular one, number amino acid down here is number 1. The final one in the end is 124 in this chain. And the chain goes like, in this case, lysine, glucine, threonine, alla, lys, B, etc. So there are 20 different possible beads on this string. Now, the whole thing doesn't look like this, of course. It really folds up in a very complex 3-dimensional structure so that a typical protein enzyme would look like this. So in this kind of representation, every atom is shown. So up here, that's a hydrogen atom and a nitrogen atom. The gray ones are the carbons and the red ones are oxygen. And there's just a couple of yellow ones in there, and they're sulfur. And they're kind of bumpy molecules. They'll often sort of looks like there's a crevice in here. Well, that might be where a chemical can fit precisely and undergo a chemical reaction. So just another way to show enzymes. Here we see every atom represented as a ball, but we just make them all the same color to simplify it. And you'll find that this particular enzyme has this pocket over here that we call the active site. And it has a shape that is precisely matched to the shape of some chemical that it is going to stimulate a reaction in. So this could be sucrose. This could be the enzyme that we call sucrase. It ends in ASE. It's an enzyme. And when it goes into this pocket, often the enzyme will change shape slightly. You see that this one closes down in here. And now in this space, a chemical reaction can occur at a much higher rate with much greater facility than it could if it was just bouncing around here in the water inside the cell. All right, so let's just look just a little bit more to pound this point home of what enzymes do. They're catalysts. They're not changed in a reaction, but they line up the molecules of other chemicals in a precise way to cause a reaction to go faster. So in our cartoon version here, let's say the red molecule has 3 atoms in it and the blue one has 3 atoms. And there's a need for a chemical reaction in which you attach one of these reds to the end of the blue. Well, what will happen is they'll bind to the active site of the enzyme. And so that will position this somewhat close. And then in the next step, for example, maybe this in blue one will actually get attached to the enzyme, but now it's much closer to the red one. And then in the next step, what will happen is that this one is modified and is attached to the, whoops, hit the wrong button here, is modified and is attached over here, and then the whole thing comes off, and now you've moved one of these molecules over here. So that's what enzymes do. So here's my summary. So almost all enzymes are proteins. They're formed from these long chains of amino acids that then fold up into a complex but precise 3-dimensional structure. Every enzyme has a different structure, a different shape. They typically have 500 to 1,000 amino acids, so they're really big, complex molecules. They function to make chemical reactions go faster. They have some site on the surface, often a little cavity. This is called the active site, and that is very specific for the types of molecules that can bind there. We have in our bodies at least 10,000 different kinds of enzymes that catalyze 10,000 different reactions. Even in the simplest kinds of molecules like fruit flies and Drosophila, and even some complex bacteria may have maybe as many as 6,000 different kinds of enzymes. So the function of the genes is in fact to encode the sequence of amino acid chains. So genes have the information to code enzymes, encode enzymes, direct the synthesis of enzymes. And lastly, they're good to eat because if you're eating plants or you're eating animal cells, what you're eating is the 10,000 different kinds of enzymes. That's the protein, a lot of the protein that is in the food that we eat. So what happens, we eat them, we take them all apart to get the individual amino acids, and then we can use those to make our own new enzymes and proteins. All right, so I'm gonna give you a chance now to ask questions. What would be best is if you use the raised hands feature with this many people. So, you know, under your participants list, you click on participants, and then you go to, there's a raised hand, and then I can see who has asked questions, and it comes up in the order in which people have asked questions. So Brian has asked a question. I'm going to unmute you. Brian, what's your question?
Yeah, I had two actually. One was when you talk about the molecules fitting the enzymes, is that really how it works, or is that just a model to describe it? I mean, is it really a physical fit process that goes on there?
It's very much a physical process because it really has to be close enough to actually change the chemical bonds between the interaction of the enzyme and the molecule that it's reacting with. And sometimes the sugar may bind to the enzyme, and then the sugar may chemically react to actually be chemically bound to the enzyme, be part of the enzyme briefly, but then the enzyme will pass it off or split it in two and release it. So yes, the interaction is at the level of atomic distances.
Okay, and then the second part was if they're catalysts and they're reusable, why do we need to eat so much?
Okay, so the lifetime of an enzyme is highly variable. So a few will really only last for a few minutes. Many of them will last for days or weeks. And so it depends on the cell, et cetera. A part of the answer is that you don't need to do all of these reactions all of the time. I mean, so after a meal, you might make a lot of these enzymes, and then when there's no food around, you might actually get rid of them. So, or anyway, it's just the kind of the economy. The cell doesn't really have enough room to store a large quantity of all 10,000 all the time. So it only makes the ones it needs at that particular time. Okay, here. All right, so that was our summary on enzymes. Now, main point of this lecture is going to be this: where do we get energy from our food? And there's a lot of energy in our food. So we talked about starches and sugars. You really want to see whether these are energetic molecules. You should remember that many people, hundreds of people, thousands of people have been killed by explosions of starch and sugars. And here's one that was caught in the act. I don't think anybody was killed in this one, but this is a grain silo. And it was overloaded in some way. It broke open. The falling material caused a spark to form. The oxygen in the air combined with the starch in the grain, and you had a huge explosion. And of course, these grain silo explosions, you know, over the years, to this day, they're probably one or two big ones somewhere. They've gotten better and better in the safety features, but it's been an old problem. But it just illustrates the fact that if you combine oxygen and sugars, you can release a lot of energy. And somehow we want to use that energy in our bodies. So how does that really work? All right, so let's go back to, we eat a potato. It's full of starch. In our intestine, the enzymes there clip off this glucose molecules in this long chain of starch. And that glucose goes from the lumen of our small intestine across the membrane of a cell, and these cells have these finger-like projections to give them a big surface area. They cross by a protein that acts sort of like an enzyme, but instead of chemically changing it, its function is to be a transporter. So it carries it across the membrane into this cell, and then once into this cell that lines your intestine, on the other side of the cell are proteins that carry the glucose out of the cell and deposit it in the blood. So take our food from our intestine to our intestinal lining, they go to the blood. Then from the blood, they circulate to our major organs, and much of our metabolism works in our liver. And so let's take this as an example of a typical cell in the liver. So via these same kind of transport proteins, the glucose that's out here can cross this membrane and go into the cell. Now, just a brief overview of the cell. We have the nucleus, that's where the DNA is, and the information to make enzymes is copied on the DNA into RNA. It goes out these pores out here. It is bound by these ribosomes, which are little protein manufacturing machines. And so you can get all of the enzymes soluble in here. Some of the enzymes go in these compartments. The little membranes that are made here are made by this reticulum of membranes here. It has among its functions to make membranes inside the cell. All right, but we've now got our glucose inside the cell. And how do we get energy from that glucose molecule? Well, this is one of my favorite slides. And the answer is, it's complicated. Okay, but what this diagram shows you, this is almost like the wiring diagram of a cell. Every dot here, black dot, is some different kind of molecule. And in this particular diagram, glucose is actually that molecule right there. Every line is an enzyme, which means that this glucose can be converted to another molecule, in this case, glucose phosphate, right here. So what it shows you is how you can interconvert all of these molecules. So some of this over here is taking the nitrogens off of amino acids and proteins, and it's how we form the urea that goes into our urine. Up here are the molecules that make the nucleotides that make DNA and RNA. Over here are molecules involved in putting things together to make fats. Other molecules are over here taking things apart to break apart fats. So this particular diagram shows about 600 different kinds of molecules and 600 enzymes to do this conversion. Certainly, a few of you have probably taken some biology, biochemistry, and have seen this kind of thing in the past. When going back 50 years, if you took a course in biochemistry, the whole course was pretty much focused on trying to memorize as much of this whole chart as you could. It was pretty dull in some ways. This sort of thing has been revived with the breakthroughs in genome sequencing, because if you sequence a genome and then you run it through the computer to find out what genes are in there, you find that most of the genes are the enzymes that are represented in this diagram. All right, but to get back to our main point, the question is, we've got glucose and we want to get energy from it. And how is that done? And that is the core of this metabolic pathway. So it shows you that it's possible to take a glucose molecule and to use parts of it to make amino acids, to make nucleotides, to make some of the components of fats, etc. But most of it we just combine with oxygen in some way and get energy, and that's the part we're going to focus on. So what we have here are 10 different molecules converted one to the other by 10 different enzymes. And this all occurs in that soupy part of the cell. And then we have this cycle down here that occurs in this specialized compartment in the mitochondria. And we're going to talk about how this works. All right, so here is the key player in all of this. And if you had to give a kind of one-word answer to how do we convert glucose into energy, that one word would be ATP. So ATP stands for adenosine triphosphate. It's a rather small molecule. See this double ring structure with nitrogens in the ring. Call that a nitrogenous base. This is a sugar, carbon with H2O attached to it, carbon with H2O to it. We got 1, 2, 3, 4, 5 carbons in this sugar. This is a sugar called ribose. I just showed you a picture of it last time. And then attached to this are these phosphate molecules. Phosphate is phosphorus plus oxygen. And this molecule, ATP, is used by hundreds, many hundreds of enzymes in the cell to provide the energy to make some reaction go, or to actually allow our muscles to expand and contract, or to move molecules across membranes, to move ions across membranes. And the energy in it is really between these phosphates. So this cartoon down here is a kind of nice way of looking at it. So you have this nitrogenous base and this sugar. And the whole function of this is to kind of be able to recognize, say, hey, I'm a molecule carrying these high energy groups on the other end here. If you bind to me, I can provide the energy for your reaction. So if these phosphates, whoops, let me go back here. If these phosphates come apart, the high energy in this bond is released and can be used to drive some other chemical reaction. Now, why is this a high energy bond? Well, there are multiple reasons, but probably one of the biggest components of the energy in this bond is something that's we can kind of intuitively understand. And that is, we know that like charges repel. Plus attracts minus, but minus repels another minus. And so these phosphate molecules have these oxygens that are carrying an extra electron, so they're all negatively charged. That means that to make this, you add a phosphate to the end of this structure here, and then if you want to add the second phosphate, somehow you've got to push this on here with this electrical charge here being repelled by this electrical charge. It's almost like there's a spring and you're compacting the spring. And then if something later on interacts with this such that that compaction energy is released, the spring part comes apart. And that's a kind of good analogy of how ATP can carry energy. And glucose, and the high energy electrons in glucose, we're somehow going to show is used to assemble these phosphates onto ATP. So just to kind of drive the point home, we need lots of ATP in our body. Some people have estimated that when we're really doing a lot of energy, we use as much as a half a pound of ATP per minute. Now, you know, if you exercise, you don't lose 30 pounds in an hour from exercising. That's because when we use it, we recycle it. We use the high energy bonds and then we remake them right away. So you start out with ATP with a high energy bond between these end phosphates. It's used for some reaction, which the phosphate comes off, and you're left with ADP, the diphosphate, just to have two. And then somehow, that we're going to go into, another phosphate gets put back on to reassemble the ATP to be reused. All right. And so we're going to focus on how to use the energy in sugar to put this phosphate back on the end of the ATP. Okay, this is our most heavy-duty slide probably for the whole course. And I'm going to spend just a little bit of time with it because you saw that diagram with the 600 different reactions. And what does it really mean? So I think if we just take some little piece of that complex diagram and look at it in just a little bit of detail, you'll see how one molecule can be converted to another molecule. Now, there's another important point that I want to make here. And that is, I said that there's a lot of energy in a glucose molecule, and you just combine this with oxygen, it can be explosive. The cell deals with this. All living organisms deal with this by taking this molecule apart just a little bit at a time, step by step. So between every one of these atoms, between the two carbon atoms, between the carbon and the hydrogen, there are shared electrons, and these electrons have energy. And so there are little packets of energy in here that we want to harvest a little piece at a time. Okay, so let's just look at what happens to glucose. The first part is just to split the molecule in half. It's a 6-carbon sugar, and you notice they've colored 3 in green here and 3 in blue here. So what you do to begin with is very counterintuitive. You want to use this to make ATP, but in fact, you start out with just the opposite. You start out by using ATP. So you take a glucose and you take an ATP, a triphosphate, and you take one of the phosphates and you put it on the end of glucose, and you got ADP, the diphosphate, left over. Now, you're going to want to put another one at the other end, but you got this double bond here. And so you slide the double bond down. So over here, there's an enzyme whose function is simply to rearrange this part of the molecule. Now, this is in a position where you can put phosphate in. So you use another ATP, you take the phosphate and you put it at the other end. And so now you've got your 6 carbons that you started out with this high energy phosphate on one end and a high energy phosphate on the other end. And you've done the whole thing to make the whole molecule more unstable so it can be split apart right here. So here's an enzyme that does exactly that. The top part becomes this molecule, 3 carbons and a phosphate. The bottom part becomes this molecule, 3 carbons and a phosphate. And then, slightly complex part here, this gets converted to this. Seeing you just kind of take this double bond here and we slide it up to the end. And so at this point, what we can say is we split our original glucose 6 molecules to form a pair of 3 molecules. And every one of these has a phosphate, a high energy phosphate on it. The next step is the most complicated of all, but really central to the whole idea of where does the energy come from. I said that these bonds connecting the hydrogens and carbons and oxygens all have high energy electrons, and these electrons can be taken off one at a time, and they are put on a molecule whose whole function is to carry high energy electrons. This molecule is called NAD. Not going to go into its structure, but just think of it, it is a carrier of higher energy electrons. And so you take a pair of electrons from here, put it on here to make this molecule here. This is going to be a central player in the making of ATP because it's carrying electrons that we can use to force phosphates to go on the end of ADP to make ATP. The other thing that happens in this reaction is that the enzyme also grabs a phosphate molecule and it sticks it onto the 3 carbons. So now, what's happened in here is we went from here to here. 2 things. 1, 2 electrons from this went into the NADH, And secondly, a phosphate went on to the other end of this. So this molecule is short 2 electrons, but it's got phosphates on either end. And so the way this whole system has evolved is we put these phosphates on here to make it more reactive, but we don't want to waste them. So we reassemble them back on to remaking the ATP. So in the next step, one of the phosphates comes off, goes on an ADP, and we make an ATP. Then we rearrange the molecule a little bit, and then we extract a water molecule from it. And the reason for this is we've got now a really high energy phosphate that we can use to make another ATP. And we come to the end of this sequence. Okay, so complicated. And this has been the bane of introductory chemistry, biology students for years, that they've often been asked to memorize all the steps in glucose. I never asked them to memorize it, but I wanted them to be able to explain it to me. But two things happen here. Glucose gets split to produce a pair of 3 carbon molecules. You used 2 ATPs at the beginning, but you get 4 of them out because every one glucose makes 2 3 carbon molecules. So you get a net synthesis of a pair of ATPs. And the most important energetically, you have a pair of these molecules, each of which are carrying high energy electrons. And we're going to use those to make a lot more ATP. Okay, so back to the cell. So all of this has occurred out here in the soupy part of the cell. But what happens next is you've got that 3 carbon molecule that's been generated, and you got the NAD carrying the electrons that's been generated. And those molecules go into this organelle right here called the mitochondrion. It is really the kind of energy generating station in the cell. These are about the size of bacteria. They are in fact old bacteria that were taken up a billion years ago and commandeered for the purpose of cells of higher organisms. Get my cursor here. All right. And a typical liver cell, this diagram is not quite accurate in its scale. A typical liver cell contains 10,000, not 2,000 mitochondria. So let's look just a little bit more closely at this important structure. So we now know that, in fact, it is the descendant of a bacterial cell from long ago. It's fairly simple in its structure in that it has this kind of soupy component inside, but then these many layers of membranes in here. And they play a key role in this converting glucose to ATP that we're going to talk about. All right, so here's what they really look like. I took this picture with the organism that Rusty and I worked with, Neurospora, and it grows very fast. And I put a colored compound in here, an enzyme that is sequestered inside the mitochondria. So this is a single cell, and it's, you know, a typical liver cell can be about this big. And so the mitochondria are like little wormy structures in there. This is about the best you can do with a light microscope to really see the details. You'd have to use an electron microscope like I showed you in the previous picture. Okay, so simple cartoon structure and emphasizes 2 points, that there's an inner compartment in here, and then there's a compartment between the outer membrane and the inner compartment here. So you can accumulate water-soluble, go back here. You can accumulate water-soluble material in this space, or you can accumulate it in this space. And that turns out to be important in our story. Okay. So one more complicated diagram here, but it's the key to the question of if you eat sugar and you breathe out CO2, where does the CO2 come from? Okay, that's one thing that this pathway illustrates. But the second is that we ended up splitting glucose into a pair of 3 carbon molecules. And those 3 carbon molecules still have almost all the high energy electrons that we want to get energy from. And how do we get those electrons in a usable form? So that occurs in this reaction that occurs inside the mitochondria. So our 3 carbon molecules come in here, and I'm not going to go through this in the same detail. The important point is that the carbon and 2 oxygens are taken apart by an enzyme in the first step here to produce a molecule of CO2, and now the molecule that is left has only got 5 carbons. And then there's another step here where this molecule with the 5 carbons, the CO2, is taken off to give you a molecule that's only got 4 carbons. And so, in fact, and then there's a third place up here where it enters the Krebs cycle, where another carbon and 2 oxygens come off to make another CO2. So shorthand of it is 6 carbon glucose. You get a pair of 3 carbon molecules. All 3 carbons in that molecule get split up to produce a molecule of CO2. So all of the glucose in the end ends up in CO2. All right, but what about the hydrogens and all the electrons? Well, you see that at several steps in this pathway, a pair of electrons is passed on to make an NADH. We saw one of these made in glycolysis, and in fact, there's NADH made here, there's made here, a molecule related NADH is made here, there's another one made here, and then there was one up here also. So at this point, once we've gone through all these steps, the original glucose has been completely taken apart, and you've seen that it takes about I think 19 different enzymes to do it, completely taken apart so that every one of the carbons is now released as CO2, goes into your bloodstream, to your lungs, you breathe it out. And many of the electrons that were in the original molecule are now being carried on this NADH molecule. Okay, so here's the summary here. Glucose, C6H12O6, goes to 6 CO2s. The hydrogen atoms go to H plus and electrons, and the electrons are being carried on this NADH. We saw some ATPs were made in glycolysis. Another pair of ATPs were made in that last reaction, which is called the Krebs cycle. But we know that glucose has enough energy to make a lot more ATPs. And we know that it does make many ATPs. So the glucose is all gone, but we still have to get a lot of energy out of it. And the energy is in these electrons here, carried in NADH. And also, where did the oxygen come into play? We haven't seen a single reaction in which anything was combined with oxygen. And that is because the oxygen hasn't come into play yet. It really comes into play in the next step. All right, I'm gonna give you another chance to ask questions here. If anybody wants to use the raise hand feature, any questions on what we went through there? I see, okay, Constantine, I'm going to unmute you. Constantine.
So this is just a practical question. As an athlete, you want to know about this cycle, and you want to know how it works.
Mm-hmm.
Is there such a thing as an athlete's diet to get the maximum amount of energy out of this process? Is there such a thing that you know of?
Well, there definitely is, and you hear about long distance runners doing carbon loading and that sort of thing. I really don't understand it. Yeah, so the answer is yes, there is such a thing, but I don't know it. So, you know, a part of it is most things that we eat end up going through the same reactions, but there are slower and faster ways to go there. And I think that's part of it. It's an availability issue. You can get energy, you can go through these pathways faster from carbohydrates than you can from fat, for example. And I think athletes, that's why they do carb loading as opposed to storing it in fat. It's more involved in getting it out of the fat cell and sending it to the liver and going through all of that physiology.
Makes sense.
All right, anybody else have another question? Another opportunity here. All right. All right. Don't be cowed by all those biochemical pathways, right? All right. I can see I can see hands here. I mean, raise your hand if you ever had to read about glycolysis. Any of you? All right. I see Paula has. All right. Anybody else? Who's raised? Yeah. Okay. A couple. Okay. All right. Okay, well, if there are no questions, then I'm going to go back to the last and most exciting part of where you get energy.
Barry, I think you have a question from Marianne. She's been
Okay.
waving,
Oh, yeah.
waving at you.
Go ahead. Oh, I've got to unmute you. Okay,
Oh.
go ahead now.
I've always wondered, because I hear people talking about the carb loading, why in the world don't they do it through proteins? Because to me, the proteins are there, they're stored, and they're still waiting for you too. Would you mind explaining that?
Well, okay, so proteins and carbohydrates have about the same amount of calories, which means energy per weight. But the proteins involve even more steps to degrade. So proteins have those nitrogens, the NHs on them.
Yes.
And that can actually become ammonia and can become toxic in the blood. So when metabolizing a protein, you've got to make sure you kind of clean up all of those amino groups and make urine from it. So the answer is it's just a more complex pathway, and it's probably not done as quickly and efficiently as with sugars. Okay. All right. So I'm going to answer these last two questions. So go back to this structure inside the mitochondrion. And so what we now have is we've got NADH carrying these high energy electrons in this space. All right, and so let's look at what's going on in this membrane, because it is the key to the energy generation in our body. So I got this cartoon from a Khan Academy lecture. I thought it was really captured a lot of complicated information in a nice diagram. So what you have here is a membrane made up of these lipid molecules. So here's the soupy part of the cell, what we call the cytosol. This is the outer part of the mitochondrion. And then inside this, there is this intermembrane space, because we saw in the structure that inside the mitochondrion is yet another set of internal membranes. And then inside these internal membranes is another space in here. Let me just make this clear. So you got the external membrane, the intermembrane space, and then the deep inside in here. All right. So what we have are the enzymes that make use of the electrons in NADH. So every glucose molecule generates 10 high energy electrons carried in by these molecules. And these electrons are used to make ATP. And this was one of the big questions in biochemistry for decades. How did this happen? And so we had seen today in glycolysis where there are enzymes that you can just extract from a cell, and you add ADP in some molecule, and the enzyme will put them together and make ATP. So it's not as though this chemistry isn't pretty straightforward. But nobody could find the enzymes inside the mitochondria that did this simple reaction. Well, it turns out that's because they don't exist, because this ATP is made in a very odd and really quite complex way. The history of this story is fascinating. For another lecture, was all figured out by a guy named Peter Mitchell in England, who basically almost did this in his garage, except his garage was in a very nice 18th century manor house in the south of England. All right, but anyway, what Peter Mitchell said, the key is we use this high energy electron to pump an H plus across a membrane. So here's an enzyme that will bind an NADH, take a higher energy electron, and the energy in the electron will be attracted to another molecule. It's going to flow downhill. And as its energy is used, it causes this protein to change shape such it picks up H plus on this side and pumps it to the other side. And it's almost literally like a little mechanical pump driven by electricity. All right. There are, in fact, 3 of these kinds of pumps, because the energy in this electron is not all used up by the first pump. There's still a fair amount left, and so it goes through a second pump, goes from relatively high level of voltage to a little lower level of voltage, and it loses that energy to pick up a proton in the enzyme on one side and move it across the membrane. And then it goes to the last one, and this electron in the end is in the end of the whole chain. What's driving the whole process is the fact that there's something at the end of the chain that has a very high affinity for electrons. It really wants to grab electrons. And what that is oxygen. This is where the oxygen comes into play. So our electrons that started out in glucose and then went to NADH and then went through these pumps, current flow through these pumps, finally end up being accepted by oxygen. Oxygen is kind of like the receiver of old, poor, worn out electrons with no energy left. And you take an oxygen atom and a minus charged electron in a plus charge, H plus, and you put them all together and you get H2O. You make water. Oxygen plus electron plus a proton, 2 protons make water. All right. So the oxygen is the driver of all of the metabolism. I mean, you really think of the reaction, the whole thing actually starts out here in the fact that oxygen wants to accept electron, and when it pulls this electron off, that leaves a space for an electron here. It gets it from its neighbor. This one then has a space for electron here. It gets it from a neighbor, and ultimately it grabs it off NADH, which ultimately grabbed it off glucose. So it's a long chain of electrons giving up energy bit by bit until they finally end up oxygen. To what end? What you've achieved here is an electrochemical gradient. That is, this side of the membrane here now is positively charged. And this side of the membrane has a lot of H's in it. This is what acid is. It's highly acidic and it's positively charged. This side of the membrane is deficient in H plus and it's negatively charged. So it's literally like a battery. Plus charge on this side, minus charge on this side. And this electrical energy in this battery is used to make ATP. And that's the last player in here. It's one of the most amazing enzymes in biology. is the enzyme that actually makes ATP using this electrical energy that's stored across this membrane. So the way it works is as the H+, the H+ over here is has a strong driving force to go in this direction because there are lots of them on this side, there are few of them on this side. So the tendency is to go from high to low concentration. Secondly, this is positively charged. This is negatively charged. Plus is attracted to minus, another driving force to go to this direction. And one way they can get across this membrane is by this channel here, which acts like a turbine. So I'm going to show you here this nice cartoon fashion. This is what the base of this ATP synthesizing enzyme looks like. Where all the protons have accumulated, there's a channel that gets them halfway through, and then they can bind to a minus charge on this protein, and it literally rotates. When this was first proposed, nobody believed this, but it turned out to be true in the end. And the protons go around on the other side, and then they get exposed to a channel where they can hop off on the inside. So high concentration flows to low concentration, sort of driving this water wheel, if you will. Plus charge attracted to negative charge, which drives this water wheel. Now, there's no ATP in here. Where does the ATP come involved? Well, that's involved in the top part of the enzyme. So as those rotating sections of the enzyme, driven by the protons moving through, go through the membrane, they cause this enzyme to rotate. And it's up here that the ATP gets synthesized. So this is looking at it from the top. But this enzyme actually makes ATP in this interface between the red part here and the green part. And what happens is that when this opens up, now ADP and phosphate goes in, then it closes on top of it. They're stuck together to make ATP. It opens and they're released. And I'm going to show you what this thing looks like from the side. So what we've got is we're not showing the turbine part here, but that part that's embedded in the membrane causes this to rotate. And this part of the enzyme here is where the ATP actually gets made. So the active site is here. It opens. ADP phosphate comes in. It closes. They're put together to make ATP. It opens, and then they pop out, and it gets reloaded. Now, the actual spinning rate here, this thing is spinning at, it does something like 400 reactions per second, and that means that it is spinning at 10,000 RPM to make ATP in your cells. Okay, so we've actually come to the end. We find that glucose and lots of sugars can be converted to glucose to be fed into this pathway. You saw in that wiring diagram that you can convert almost anything to anything else. It's combined with oxygen, and that has a net production of 6 CO2s, and the 6 carbons here combine with these 6 carbons in the glucose. And where the other 6 carbons come from, because 2 times 6 is 12, they actually come from water molecules that come into the reaction. These oxygens here, on the other hand, are binding electrons and then combining with H+ to make water, H2O. And this whole process is generated, let me say this again, this whole process is used to drive this gradient of H+ across this membrane inside the mitochondrion. And that gradient in turn drives that turbine-like enzyme that puts ADP and phosphate together to make ATP. And for every 1 glucose molecule, you make 36 of these ATPs. And what happens to them? Well, they bind to the muscle, the enzymes in your muscles, cause your muscle contraction. They allow you to synthesize proteins and nucleotides and all the components of your cells. They allow transport to occur. It turns out that your major use of ATP in your body uses something in the order of 40 to 45% of all the ATP is to keep the sodium and potassium balance right in the blood in your cells. It pumps potassium into your cells and sodium out of your cells. And that's so important because that's what drives your nervous system. The moving of these charged particles across the membrane is the basis of the electrical system that drives your nervous system in your brain. Okay, so I think we're going to stop there. And I don't know if anybody has raised hands questions, or if you actually physically raise your hand, I'll try and call on you too. The problem is, of course, I can only see a few of, a third of your pictures at a time. Any questions out there? Okay. Now I'm going to send you all the slides like I did from the last time, so you can spend your time working through glycolysis and the Krebs cycle and figuring out where all the protons go, and because it's going to be really important when we do the exam, that you'll be able to describe the third step in glycolysis. Okay, so I got a question from Gail. I'm going to unmute you. Gail, go ahead.
I still don't quite understand where the energy came out, where you got that energy to move your muscles.
Okay, so the energy ultimately is in the electrons in glucose.
Okay.
And that energy is used to make the ATP. I think the best way to think about it is the energy is you force these two phosphates together, which are normally repelling each other because they're both negatively charged. And so it's like you cocked a spring. And so when you contract your muscle, they grab one of these ATP molecules and they put it in a position such that it splits. And as they come apart, they actually cause the molecules in your muscle fibers to walk along the muscle fiber. So ultimately, it's the glucose molecules, electrons, are used to make the ATP. And the ATP is what's the direct fuel for all of these other reactions.
So does the ATP cause the break that releases the energy?
The break in what?
Well, you said there was some kind of, these two things have been put together.
Yeah.
That have been forced together, and something breaks them apart that gives that burst of energy. I mean,
yes, and that's something that breaks them apart. There are hundreds of different enzymes which have on them a site in which you can kind of load ATP in there, and then under the appropriate trigger, that ATP will come apart. And that then can say like make a little muscle protein move, or cause two proteins to be synthesized to stick together. It's the ATP binding and the energy and the kind of spring-like mechanism between the phosphates that can be released and directly linked to some other chemical reaction.
Okay, thanks.
Okay, let's see. Okay, and then Constantine, I'm gonna unmute you. Why? Okay, for some reason.
Myself. There we go.
Okay, there you go. Okay, yeah.
So on the same line then, what causes some bodies to have a higher reflex, which is, you know, the idea of an athlete, how fast their muscles can react. Is it the same in all? Obviously, it's not the same in all human beings, but does that, for instance, is that motor device constant in the human beings? And so the question is, what gives some of us more reaction time, in other words, a better reaction time than others?
Okay. Well, yeah, well, I mean, that's quite a ways from things I know about, but I think the short answer is that's the inherent difference in the activity of the molecules at the level of muscle is probably not different. That is probably all the wiring of the nerves. In other words, I mean, reaction time is just developing a very fast nerve signal from, for example, what you see, you're trying to hit a baseball at 100 miles an hour.
Right.
It's a neurological thing rather than an underlying biochemical.
Ah, okay.
All right. Actually, there was one other question earlier. Alexandra, did you have a question? Yes, I'm going to unmute you. Okay. And if I can't do it, that often means that you've muted yourself. Okay. Good. Okay, Alex.
Say something?
Yeah. Okay.
Can you hear me? I just wanted to, exercise physiology was my master's degree.
Okay.
And when you talk about reaction time, that gets to be really complicated because there is the nervous part. And then there's the fast twitch versus the slow twitch muscles, which are just like, you know, when you eat a chicken, there's white meat and there's dark meat. And your white meat tends to be the fast twitch and your dark meat is the slow twitch, and they're closer to the bone. So in an animal model like a frog, you can pretty much predict by the species what the percentage is going to be per limb. You take a thigh and you pretty much know, whereas in human beings, it's very different. And the only way you can really tell with human beings is they do these muscle biopsies, which are rather painful. You can pull it out and say, oh, he's got greater potential because he's got more fast twitch. And that was quite a field for a while. And then I also wanted to say, in terms of people eating protein for exercise, there is a big field of research on post-exercise drinks, because that's when you want the protein. And they claim the first half hour after you've done really heavy exercise is the most important. And every minute counts. So when you get in, you should really be having your protein like right away.
Okay, interesting. Okay.
Yes.
Anybody else? Okay. Anybody else? Oh, who I got here? Okay. Joanne, I'm going to unmute you. Okay. Okay, go ahead.
Yeah, Barry, mine goes back to your opening comments, if I might. You told us about the woman who has a history in the coronavirus research, and you said UC crawl or something, and I couldn't catch the word that you said if we wanted to look it up.
It's called the Craw Lectures, K-R-A-W. And
K-R-A-W,
yeah, they happen once a month over in Silicon Valley, and it's kind of a big deal. They really try and get some star from the university to talk to the Silicon Valley people. And this was so timely. And I don't know, Benita said she heard it, said it was really good. Maybe some other people did too. Yeah.
Is that something one can still?
I see Nancy maybe. Okay, yeah.
Is it something we can still find online to watch?
They do a real professional recording of it, and whether it's there yet, I'm not sure, but it's likely to be within a day or two. And you should be able to just look it up. Her name was Rebecca Dubois, D-U-B-O-I-S.
Oh, right. That's great. Yeah. And then just go online to the Crawl Lecture Series, UC.
Yeah, UCS.
Okay, great.
Yep.
Great, UCS. Okay.
Okay, and
Thanks, Barry.
And Benita.
I just wanted to say they told me it would be on YouTube.
Oh, yeah.
That it would be on the YouTube channel. So That's where you might find it, but I don't think it's going to happen immediately.
Oh, thank you.
Sure.
It's good to know. Appreciate it.
Welcome.
Okay, anybody else?
Yeah, can you hear me?
Yes.
Hi. I noticed that in the newsletter coming up for the fall semester that you're teaching a couple of classes. And I was wondering why it appears that one of them is your nutrition class. Is that the same one we're taking now?
Well, the newsletter, I think what it said, it was the fact that my class was one of the ones interrupted by the shutdown, and that maybe we would resume it in the fall. But since I've resumed it now, I won't be repeating this in the fall. But I have for many years done this topics course on molecular biology, and it's certainly my intention now to do that again in October. The way that usually happens is I'll do an intro lecture, basically what are genes, what are proteins, etc. And I will also do a lecture on some topic, and then I invite three of my colleagues. Try to rotate it from year to year. Might talk about stem cells or neurofunction or eyeball generation or whatever. I invite three colleagues, and they each give a seminar. So that'll probably happen in October. Very likely in a Zoom format. We'll see.
Great. Thanks for answering my question.
Okay. Anybody else? All right. Well, okay. I hope that wasn't. As I said, that was the most biochemical lecture, and I'm going to send you the slides. And next time, I think what we're going to talk about first of all is I just mentioned how oxygen drives the whole thing. Oxygen is really a pretty toxic compound in some way. And so we're going to talk about at the beginning, what are antioxidants? They're supposed to be really good for us, but what are they? They're not really good for us. And the answer is, as to so many things, it depends. All right. Okay, so I will see you all in a week then. Thank you very much.
Thank you, Barry.